Welcome to a special edition of Life and Biology. I'm Dr. Joel Graff, and it is both St. Patrick's Day and a quarantine day. But that's all I'm going to say about coronavirus, because I want to tell you about this really cool new study that came out and found T cells that could go and attack lots of different types of cancer. So I want to explain what that study was about and what it might mean. So I've got a couple different cells drawn here. Usually I draw a cell as a circle, but I'm zooming in on an interaction between these cells, and I have the proteins that are interacting between the cells kind of drawn with a little bit of detail. Over on this side, I have a T cell, and on this side, I have a cancer cell. This T cell happens to be a CD8 positive T cell. This protein here is called CD8, and CD8 positive T cells can be cytotoxic, which means that they can go and attack other cells. So that's where I got the title "Killing It" on this slide. Still working on trying to get comfortable in this situation, but anyway, we've got the CD8 T cell here. T cells have T cell receptors. That's what the TCR stands for. And in orange here, I've drawn out the T cell receptor. It has an alpha chain and a beta chain, meaning it's like two proteins working together as one. And then on the cancer cell, across from the T cell receptor, I have an MHC1 molecule, major histocompatibility complex 1. So we'll just call it MHC1. This protein also consists of two polypeptides. One is the alpha chain of that protein, and then there's a little protein here just kind of floating there. What its purpose is to hold the structure of MHC1, and it's called beta-2 microglobulin, or B2M. So the important part of the MHC molecule is there's a couple important things. One, the T cell receptor can recognize MHC1. The CD8 protein can also recognize MHC1. And then there is part of the T cell, the very tip of the T cell receptor, is interacting with both the MHC molecule and that red dot that I have in there. That is called the antigen. And so this is a specific peptide, a series of amino acids, usually 8 to 10 amino acids for CD8. T cell, and it is what the T cell receptor was, or it's what the T cell was born to recognize. Now this is a cancer cell, which if we're talking about your own T cell trying to attack your own cancer cell, usually you think that wouldn't happen because you would not want your own immune system fighting itself. However, certain types of cancers, let's say melanoma or those related to smoking, have a large number of mutations in their genome, and if those mutations occur in a protein coding sequence, then the protein that that cancer cell is making is going to look slightly different than that same protein elsewhere in the body, because there's mutations to the genome of the cancer cell. So what happens, and I won't get into the detail of antigen processing and presentation, but this protein gets chewed up into peptides, and then this dot here is supposed to represent a peptide that formed when a different neoantigen like this was chewed up. Okay? So what happens when a T cell recognizes a cancer cell and sees that neoantigen? Well, there's a number of molecules that the CD8 T cell will secrete. Some of those molecules will poke holes in the cancer cell. Other molecules are proteases. We learned about proteases earlier, and they will get into the cancer cell and start chewing up things within the cancer cell, and then eventually cause the cancer cell to die. So again, it throws out some proteins called perforins that'll poke holes in the cancer cell, and then it throws out some proteases called granzymes that will get into the cancer cell to kill it. Okay, well, for a T cell to kill a cancer cell, it needs to be activated. So this next slide says signs, everywhere there's signs. And there's three big signs that let you know that a T cell has been fully activated. The first sign is proliferation. So one thing about your immune system is that all the different T cells are unique in the T cell receptor they make. However, if you find a T cell that would be helpful for killing, say, a cancer cell, it would be good if that T cell replicated. That's called proliferation, the replication of that T cell. And if one cell becomes two, becomes four, as I've kind of drawn down here, you will pretty soon end up with a clonal population. Clones meaning a bunch of copies made from an original. Okay, and I'm going to come back to that when I talk about the CFSE assay at the bottom. But one is that cells will proliferate. Another is that those T cells, in addition to making those perforin and granzyme molecules, will also start making things like cytokines. And in this paper that I'm going to link to, they were looking for the production of TNF-alpha. TNF-alpha is an example of a cytokine that's secreted from one cell and can act on another cell. Cytotoxic activity is the third, so can it go ahead and kill that other cell? Meaning if you put these T cells in the same dish as cancer cells, would these T cells kill the cancer cells? So throughout the paper, and I'm not going to go figure by figure walking through the paper, but throughout the paper they did a number of different assays to look for proliferation, or to look for cytokine production, or to look for cytotoxic activity. I'm just going to highlight one. So in the very first experiment of the paper that I'm describing, they took some cancer cells and they took some T cells, and it wasn't just one of each. They had a whole population of T cells. The cancer cells were all identical because they were copies of each other. But they were asking, were there any T cells in the population that could recognize those cancer cells? And the first way to know that a T cell could recognize antigen and recognize a cancer cell is that this T cell would replicate. So I've filled in this T cell with a green color. The stain is called CFSE. That's where this assay gets its name. But CFSE is added to the T cells when they first go into the well with the cancer cells. And so the T cells all start out being bright. So we'll say 100% green color. When that first T cell divides, this one daughter cell gets half of the CFSE, and the other daughter cell gets half of the CFSE. And so if you had a way to measure the amount of brightness of green of those cells, it would be exactly half, or roughly half. Let them divide again so that you go from 1 cell to 2 cell to 4 cells. Now your denominator is 4, and they're going to be 1/4 as bright as your original T cells that you put in. Give it a while to go, and if these original T cells are super bright, if they multiply several times, let's say 8 times, that's 2 to the 8th, which I think is 256, it's going to be 1/250th as bright as the original cell. And so that's what they did in this paper, is they took these T cells, put them in with cancer cells, waited a few days, and then asked, did any of the cells expand? Did any of those cells recognize the cancer cell and become activated? So they did find some cells that would replicate when they saw the T cell, and they managed to sort the cells out, 1 cell per well in a plate, so that when each of those wells, a cell in each well would replicate, you'd end up with a clone. And then they screened those clones for their ability to see the cancer cell. Now, on the previous picture, I said that an alpha beta T cell on CD8 positive an alpha beta T cell receptor on a CD8 T cell would recognize MHC1. But in this paper, what they did, and they used a CRISPR screen, and they just knocked out all sorts of genes and asked what gene can we knock out to prevent the T cell from killing the cancer cells. But when they did that screen, they came up with this hit in their screen called MR1. Well, MR1 is a protein that looks very similar to that MHC1 protein we saw in the first slide. In fact, it even shares the beta-2 microglobulin subunit here, but then this alpha chain, I don't even know if they call it an alpha chain, but what amounted to the alpha chain on the MHC1 molecule is instead MR1 sequence. And then instead of the red dot indicating a peptide in the antigen binding spot on the antigen presenting molecule, they found that it was actually a product of metabolizing vitamin B. Okay, so I'll step back and say usually when a CD8 T cell recognizes an antigen, it's a peptide. But these molecules, like MR1, there's a few others like CD1, they can put antigens that are not peptides, not fragments of proteins out there. It was known that this MR1 molecule could see antigen in a, it could see antigen when enzymes from bacteria change the form of vitamin B to some other type of, it's called a metabolite, and then that metabolite would end up there. But this T cell receptor didn't see the type of metabolites that you would get from bacteria acting on vitamin B. And so it seems like maybe the enzyme that's acting on vitamin B to produce this molecule that's out in the MR1 molecule and is the antigen that this T cell sees, it seems like maybe it's something that the cancer cell itself is doing. And what's exciting about that is that they originally started with some lung cancer cells, and they found that this T cell could kill the lung cancer cells. Well, then they started putting these T cells in dishes with cancer cells of other tissue types, and it would kill the cancer cells. So that's great. It seems to kill all these different cancer cells, but what about, would it kill a normal cell? If they put these T cells with this particular alpha beta T cell receptor in with a normal cell, a non-cancerous cell, the cells would not get killed by the T cell. So now we've got a T cell that doesn't seem to kill a wide variety of normal cell types, but it does kill a wide variety of cancer cell types. And they followed up with several more rounds of experiments. They expanded the number of cancer cell lines they screened. They expanded the number of normal cells, and the pattern held. These T cells like to kill cancer cells and all sorts of different types of cancer cells. So in kind of the final hurrah of the paper, they took the information that encodes that T cell receptor, and they took the DNA sequence and they put it into a completely different CD8 T cell, and they had this completely other CD8 T cell that made its own alpha beta receptor, but they made it overexpress this particular sequence of T cell receptor that this clone had, and it was able to cause the person's regular T cells to attack cancer cells, a number of different, say, melanoma cell lines. And that means, with any luck, we can take this information about this particular T cell clone, have our own, if I were to get cancer, it's possible that someday in the future, after all the safety tests and all that have been done, it's possible that someone could take some CD8 T cells just right out of my blood, manipulate the cells a little bit to have them express this particular T cell receptor, and then put them back in my body, and those T cells could potentially kill my cancer. And so this is exciting because most other CD8 T cells, they will recognize, if you remember, it was a specific type of neoantigen. Well, if one cancer makes one type of neoantigen and a different cancer makes another neoantigen, then this peptide is going to be different, and whatever T cell receptor was here wouldn't be able to recognize all those new cancers. But this new study, it provides hope that this new T cell receptor could potentially be used as a universal receptor that you could add to any T cells, CD8 T cells of any person, and attack any type of cancer. There's a lot of work to be done to make sure that this sort of thing can happen, but it would be great if in a few years we look back and take a look at this study as the beginning of the end of our war on cancer. All right, have a good day. Like and subscribe or not.